Zoonoses have large impact on the most populous countries, which are usually under high environmental and economic pressure. In these places, the contact with wild animals is frequent. Although the contact with wild animals is less frequent than the contact with domesticated animals, when pathogens are transmitted to humans, the severity of diseases is disproportionately higher, precisely due to the absence of previous contact. Zoonotic pathogens reach humans mainly through hunting or consumption of wild animals. Some of these pathogens can later be transmitted from person to person. The relationships between hosts of different taxonomic groups, given by the pathogens transmitted among themselves, are represented in this core chart. When we look at human relationships with other hosts, the edge with domesticated animals is the most frequent. There are over 1,400 transmissible diseases described in humans. Most of these diseases are caused by zoonotic pathogens, or 62%, due to our close relationship with domesticated animals by consumption, use of byproducts, or by keeping them as pets. Among domesticated animals, especially those closest to us like ungulates or carnivores, the proportion of pathogens transmitted between different taxa, including humans, is very high. If we consider only the emerging diseases, the proportion of zoonotic pathogens involved is even higher, or 75%. Despite the low proportion of the viruses as human pathogens, or 15%, its association with emerging diseases is disproportionate. Zoonotic viruses are the most frequent emerging pathogens. Of these, 90% are RNA viruses and 91% transmitted by wild animals. These viruses generally have a wide range of hosts, jumping to human mainly through hunting and consumption. This is the list of re-emerging and emerging outbreaks reported to WHO over the last 20 years. All of them were caused by viruses. Those diseases marked in bold captions are caused by RNA viruses, and those with asterisks are transmitted by bats. Note that only one, monkeypox, was caused by a DNA virus and all other caused by RNA viruses. Mammals are the most important reservoirs of zoonotic viruses. There is a positive correlation between diversity of mammals and zoonotic viruses, as shown in the figure. In the descending sequence, rodents, bats, carnivores, and primates, the most diverse mammalian orders, also present the most diverse zoonotic viral richness. Rodents are reservoirs of adenovirus, for example Lassa, and Rna virus, for example hantavirus and Crimean Congo hemorrhagic fever virus. And bats are reservoirs of paramyxovirus, for example Hendra and Nipah, filovirus, for example Ebola and Marburg, Lissa virus, for example rabies, and coronavirus, for example SARS-CoV-1, 2, and MERS-CoV. Despite rodents are the reservoirs of the highest diversity of zoonotic viruses, bats are the reservoirs of the highest diversity of emerging viruses. There are 27 mammalian orders, 1,214 genera, and 6,495 known species. Here the 27 mammalian orders are organized by genera, in chart A, and species, chart B. In both charts, rodents make up 39%. Bats, the second more numerous mammalian order, make up 17% of the genera, or 227 genera, and 21% of the known species, or 1,386 species. Bats have different sizes and shapes. The biggest bat is the flying fox, weighing 1,600 grams, with a wingspan of 1.5 meter, and the smallest is the Kitti's hog-nosed bat, weighing only 2 grams, with a wingspan of 0.3 meter. Bats also have different feeding habits, from frugivorous, insectivorous, nectivorous, carnivores, and even hematophagous. But most importantly, bats are the only mammals that take flight, which is a key feature in their role as RNA virus reservoirs. The association of bats with RNA viruses is the result of a long coevolution. This association is the result of several factors. The great diversity and distribution of bats offer viruses a large pool of hosts. In addition, bats' adaptation to flight makes their immune system more complacent to inflammatory processes. The flight capacity itself is a great selective pressure upon viruses, which partly explains the long coevolution. In addition, migratory and dispersive capacity resulting from the flight has a relevance in the transmission of viruses by bats. Thus, Lissa virus, for example rabies, Henipavirus, for example Nipah and Hendra, Filovirus, for example Marburg, Ebola and Mengele, and coronaviruses, SARS-CoV-1 and 2 and MERS-CoV, all RNA viruses, have bats as hosts. Reservoir species often display no symptoms, as the host and pathogens are well adapted to each other. However, when there is spillover of these pathogens from animal reservoirs to humans, the effect can be devastating. Zoonoses are especially problematic to humans because our immune system has not co-evolved with those pathogens. Bats are reservoirs to a number of viruses, especially RNA viruses. This figure shows some examples of RNA viruses that jump to humans, sometimes using an intermediate host for transmission. Now I'm going to narrow the discussion using coronaviruses as example. According to a report of a WHO mission to China released last month, 4 hypotheses of the origin of the new coronavirus have been investigated. The first one was related to the introduction of the virus through direct zoonotic spillover, represented by the red arrows. According to this hypothesis, bats or other animal infected humans directly, and afterwards human-to-human transmission would have occurred. This hypothesis was considered likely. The second hypothesis was related to the introduction of the virus through zoonotic spillover with an intermediate host in the transmission. According to this hypothesis, bats or another animal infected the intermediate host, which infected humans, and afterwards human-to-human transmission would have occurred. This hypothesis was considered very likely. The third hypothesis was related to the introduction of the virus through food products. This hypothesis was considered unlikely. The fourth hypothesis was related to the introduction of the virus through laboratory accident. This hypothesis was also considered unlikely. Bats are not the only ones responsible for their role as reservoirs, but the viruses themselves. Coronaviruses are great examples. The large coronavirus genome, resulting from the acquisition of genes encoding RNA processing enzymes, the frequent nucleotide recombination, shift, insertion, and deletion, and consequently their great genetic plasticity, are responsible for the possibility of transmission between different species and its adaptation to new hosts. This is why all human coronaviruses are of animal origin, and the taxonomy of coronaviruses is constantly changing. This phylogenetic tree of the alpha and beta coronavirus genera isolated from bats until 2019, shows that beta coronavirus will be the ancestral genus and the nobecovirus, or lineage D, the probable ancestral subgenus. Despite the great importance of bats as hosts of coronavirus, few scientific studies have been carried out to determine the global circulation of these viruses in bats. However, both alpha and beta coronavirus are cosmopolitan. In beta coronavirus genus, however, the distribution of sabecovirus isolates tend to be more frequent in the Old World, whereas of merbecorona more cosmopolitan. Nobecovirus, in turn, tend to be restricted to the Old World, especially Southeast Asia. China is the country with the highest number of coronavirus isolates in bats. The circulation of coronavirus in bats is well documented in China. The distribution of coronavirus in bats is shown in this map from a literature review published in February 2019. There is a great diversity of bats and coronaviruses among Chinese provinces, especially in the southeast of the country. For example, in Guangdong province, painted in dark gray, where SARS-CoV-1 isolated in the bat genus Rhinolophus appeared. The province of Hubei, where SARS-CoV-2 appeared in December 2019, is also highlighted in the map. In addition, China is the third country in area and the most populous in the world. Due to its great extension, it presents several biomes, resulting in a diverse chiropterofauna. Nevertheless, as 2020, 23 of the 39 species of coronavirus has been discovered in China. In this literature review, the imminence of new strains of coronavirus related to SARS-CoV was stated. This phylogenetic analysis of alpha and beta coronaviruses, published in August 2020, shows the ancestral bat families that host coronaviruses. For both alpha and beta coronavirus, the ancestral host families are Rhinolophidae and Vespertilionidae. Beta coronavirus are family specific, with the sabecovirus found mainly in bats of the Rhinolophidae family, the merbecoviruses in Vespertilionidae, the ibecoviruses in Hippocyderidae, and nobecoviruses in Pteropodidae. Sarbecovirus and merbecoviruses have proven zoonotic characteristics. Nobecoviruses, although found only in Pteropodidae, restricted to Southeast Asia, have high zoonotic potential and must be carefully monitored. Bats in this family are host to several zoonotic RNA viruses such as Ebola, Hendra, Marburg, Nipah, and orthoreovirus. A nobecovirus recombination event with orthoreovirus, identified in 2016, raises the hypothesis of a possible recombination with other zoonotic RNA viruses identified in bats of the family Pteropodidae. Bats of the family Rhinolophidae, preferred hosts of Sarbecovirus, are found in the Old World with a wide distribution from West Africa to the Oceania. Despite this is not the area of the greatest bat richness in the world. The Neotropical region is the richest in chiropterofauna in the world, especially in the Amazon. In this work published in 2018, we observed that in some regions of the Amazon, more than 100 different species of bats can be found in a single location. It is also noted that in Southwest Asia, although quite rich in chiropterofauna, does not compare to the Amazon. This demands an increased surveillance of bats not only in the Amazon, but also in Brazilian rainforest. This need is reiterated by this work published in 2016. Map A shows the known richness of virus transmitted between bats and humans, where the greatest richness is observed in Africa. Through mathematical models for predicting viral richness observed in map B, the greatest richness of viruses transmitted between bats and humans is the neotropical region, demonstrating a failure in bat virus surveillance, especially in Brazil. Finally, based on the distribution of the human population in their habits, we observe in map C that the regions where there is a greater probability of transmission of virus from bats to humans are China and India, followed by sub-Saharan Africa, Europe, and Brazilian rainforest. This is a reminder of the huge public health challenge that we will still face in the future. Despite all that has been said, bats are not to blame for the emergence of viruses. The reasons for the emergence of viruses are deforestation, hunting, and consumption of free-living animals, wild animal trafficking, among others. Transmission to humans can occur at precisely those occasions. A guide to communicating about bats to avoid their persecution during the COVID-19 pandemic was published in 2020 with several points of attention. First, the need to expose fake news about the culling of bats, avoiding repeating a myth that increases its familiarity, reinforcing it. Correct communication involves saying that culling of bats further increases the risk of zoonosis, something similar to what we saw in the yellow fever epidemic in southeastern Brazil between 2016 and 2018 in relation to monkeys. In addition, negative effects must be neutralized, which leads to undue fear of bats, a common behavior in Latin America due to rabies transmitted by bats. Thus, assisting to injured bats should be encouraged, avoiding direct contact, of course, and calling responsible authorities to emphasize the fact that bats are the largest seeds dispersers. Finally, support of environmental norms should be encouraged, emphasizing the importance given to bats and the benefits of their presence. I hope you are all interested in studying bats and their pathogens now, am I right? Thank you for your time and hope you are all safe.